Okay, good morning, students.
Good morning, sir.
Yeah,
Morning, sir.
so
Good
today
morning,
we'll
sir.
today. Good morning, everybody. Today we'll continue whatever I have discussed yesterday. So from there we'll continue about different kind of biological networks. And next week there will be holidays, so I think Wednesday they will declare holiday after Ayush Puja, and 14th is again Ayush Puja. So next week there won't be any class. So we'll meet. Next class will be
Sir,
tell me.
sir, can we have the internal one on 20th October, Wednesday, after the next week?
Yeah, fine, no problem. No problem.
Sir, and what would be the syllabus, sir, for the test?
Syllabus we will have whatever I have covered, no? Whatever I have covered before, not you remove biological network. Before that, whatever I have done means different kind of scale-free network, random network, then introduction to systems biology, everything will come, small world. Okay?
Okay, sir, so before biological networks, like unit 1 and 2, right?
Yeah. So it is 20th.
Yes, sir, Wednesday.
20th we want to do. No problem. We will conduct. So basically we are meeting for exam only. Okay, good. So 20th there will be exam, and there won't be any class on 13th and 14th. Okay? Yeah, 14th is
Okay,
holiday.
sir.
13th also they will declare holiday after Ayush Puja. Okay, all departments here perform puja. Okay? So we will meet on 20th for the exam. Okay, I think I will. You have two classes, so I will start around 11 exam. Okay, so try to join that in 50 years. Yeah. So today I will continue whatever I have discussed in my last class. So I was talking about So I was talking about different methods how to make a gene regulatory network. So one aspect I told that you can do using electrophoretic mobility shift assay. You can also do using footprinting assay. This is called deoxyribose footprinting assay. Here, deoxyribonucleases are they cut the DNA into shorter strand, and there are many DNAs which cut the DNA at particular place of the sequence. But footprinting technique uses relatively indiscriminate DNAs that will cut DNA at any point. If a protein binds to DNA at a particular location, then it will not allow DNAs from cutting the DNA or close to the location. So footprinting will help us in by mixing strand of DNA containing the sequence to be tested with DNAs and observing the mix of strand length after DNase has cut the DNA sample into pieces in a variety of different ways. So you can repeat the experiment many times, and protein present will result in different mix of strand length if the protein binds to DNA and prevent it from being cut at certain places. The mix is usually determined using a gel electrophoresis method. So both mobility shift and footprinting assay tells us that where protein binds in the DNA. Okay, and if you want to pin down where it binds, typically you have to do further work. You can create many oligonucleotides, and then you have to see that where the protein will bind. So now once you know the gene, that is a function like a regulatory gene. So for that, if you want to verify that a particular protein is a transcription factor or not, you can do it computationally or you can do experimentally. The computation approach is determining whether the subsequence in which protein binds is indeed a promoter region for the identified gene. And this is substantially harder task than simply identifying nearby gene. The structure of the promoter region, unfortunately, quite complex and varies widely, but there are computational algorithms to identify. So you can perform your experiment to measure directly the concentration of messenger RNA that through DNA microarray, and then you know that DNA microarray depends on the fluorescence technique. So there are two dyes, Psi3 and Psi5, and based on their expression, you will be able to tell the expression of the genes. So based on this information, you can also develop the gene regulatory network. Now there is another type of network which is known as neural network, where you know that our body, brain and central nervous system plays a very important role. And the main function of the brain is to process the information. And this primary information processing is performed in neuron, which combines several inputs to generate a single output. So our brain has billions of neurons which are wired together. And so we have a neural network in our body, in our brain. Now, you know about this structure of the neurons. I'm not going into detail. That's synapse and dendrites and all these structures. So if you talk about neural network, in the simplest level, we can say that it's a unit that accepts a number of input, either the input may be excitatory or inhibitory, and combining them and generate an output result that is sent to one or more neurons. In a network term, a neural network can be represented as a set of vertices, that is the neurons. The vertices are here neurons, and they are connected by two types of directed edges. One is excitatory and another is inhibitory. So you can, by convention, you can show the excitatory one by arrow, and inhibitory you can show by the that edge ending with bar. So in practice, neurons are not same. They are of different type of neurons, and so you can determine the structure of neuron through microscopy, like electron microscope or optical microscope. So a wiring diagram of a neural network is important. This is the example of a wiring diagram, brain circuitry of a C. elegans worm, because you know they have a fixed number of neurons. So this diagram is made with the help of electron micrograph of a worm brain, and then this is the C. elegans is the best studied organism. And the brain of C. elegans, simply, it is less than 300 neurons, and every specimen of the worm has the same wiring pattern. So there are different types of neuron denoted by shapes and level, along with number of different type of connection, both excitatory and inhibitory. Some of the connection run out of the figure or enter from somewhere of the pages. There are connections that run through another part of the network also is there. So this is the part of a brain circuitry. It's not full. Okay. So this neural network of C. elegans is also important. And then you have some networks like ecological network, if you look at the environmental aspect. So ecological network, you can think of food wave, food chain, all these things are part of the ecological network. So food wave is a very important network in ecology. So here it's a directed network that represents the species prey on which others of a given system. The vertices of the network respond to species and directed edges of predator-prey interactions. And you can see the directed edges here, for example, who is feeding on whom, and based on that, you can make a directed network. This is the food wave species of food wave of species in Antarctica. Okay. So the direction of edges in the network. Come, sir. Yeah, sit down. 2 minutes. I'm taking class. Can you wait for 2 minutes? I need to talk to my colleague. Okay. Yeah. Just wait 2 minutes. Yeah. So yeah. So what happens? We see the direction of edges in the network, and the edges would point from predator to prey, but ecological conventionally draw them to opposite direction from prey to predator. So A is representing the eating of fish by birds, run from fish vertex to bird vertex. The reason for apparently this choice is that the flow of energy within the, it shows the flow of energy within the ecosystem. So almost all arrows in the figure, so directed network with this property are called acyclic networks. And so the acyclic nature of food wave indicates that there is an intrinsic pecking order among species in the ecosystem. So you can, this trophic level, you can understand using this ecological network. Okay. So food web is one example, and there will be also some other kind of ecological network like host parasite network, where you can connect the network of parasitic relationship between organisms. So how a particular large-bodied animal and the insects, they are connected with microorganisms that live on and inside it. So parasitic relations are important here. So one species is eating another, and therefore the parasitic interactions do form networks that are somewhat similar to traditional food web. So parasites themselves frequently play host to still a smaller parasite, which may have its own still a smaller one. So you can use this network between the parasite and the host. Even sometimes hyperparasites are parasitic on parasites. Okay. So this kind of relationship also you can infer. And then also this book has very nice presentation of this adjacency matrix and network and weighted network, directed network, all the concepts. Okay. So you can read from here. He has explained in a very simple way. So event degree, path, okay, geodesic path, and Eulerian Hamiltonian path, then different components of directed network, then independent path connectivity and cut sets. Everything is nicely explained. Okay. So you can read this, and you also read the different measures like degree centrality. So you know there are different types of centrality measures. One is the degree centrality, which is the simplest one. Then there will be a centrality called eigenvector centrality, which based on their eigenvector values, And also you have other centrality like cause centrality, that is not so important. Page rank is important for the websites, and so hubs. There is the idea about hubs, what are hubs, and how hubs are important. And then you have closeness centrality, and betweenness centrality is also very important. So hub centrality, nowadays people are preferring both hub centrality as well as the betweenness centrality. So betweenness centrality is the centrality which measures the extent to which a vertex lies on paths between the two vertices. Okay, so how much it helps in connectivity in the network. That's why that is between centrality is also important. Okay, so you have to study this one also. And I told you there is also a term called transitivity. This is very important in social network because how much, so it is just the transitivity is like a clustering coefficient. Okay, but transitivity word is being used for the whole network. It's a global property. Transitivity is a global property. Okay? And there is also local clustering coefficient, which is for the particular node or particular vertex. So this is what is called local clustering coefficient. Okay. So all these things you can study from this book as well. Okay. And I have also told about assortive mixing and disassortive mixing. So I do remember that in the last class I talked about assortive one. So it depends on the degree of the network. So if the generally high degree nodes don't mix with the other high degree nodes. So this is called disassortive nature. So a network is disassortive mixed by degree. Then high degree vertices tend to connect it to low degree one, creating a star-like feature in the network that are often readily visible. So disassortivity network don't usually have a core or periphery split, but they are more uniform. Okay. And assortive one is not that common in case of biological network. It may be true in case of social network. Okay. So you can here some concept of power law and scale-free network is there. You can read this part. Okay. So if you read this part, you will have a very good idea, not only about different type of biological network, but different measures of networks and how they are being performed. So I think this is enough if you read this much of information. I hope you might have got this book. Okay. So you follow this book and read these parts. In addition to what I have given from the book of Bharavasi. Okay. Any questions you have? So biological networks I have completed. Now I have already shared with you my review paper on computational medicine. I will also one day I will explain you different concepts that I have written in the review paper. So that we will discuss some other day. Okay. If you have any queries, you can ask. Who is present today? Amal is present. Arnab is present. Today Bablu is absent. Chandan is present. Chandini is present. Vidya is present. Hema is present. Mathangi is present. Then Megha is present. Naurasi is present. Nishi is present. Nishi is absent today. Sadhya is present. Satish is present. Vivas is present. And then Vishal is present. Today Uday Anand is also absent. Shubhashish is absent today. That's what. So we will discuss computational medicine maybe in another class. But next class, that is on 20th, we are going to have, you just inform your friends who are absent today about the exam. Okay? Who will take the responsibility? Hema, will you,
Yes, sir.
you explain to just inform
Yes,
them
sir. I will inform them.
the exam. And I'll be connecting through our old link only. Okay, they are changing some setup classroom, but I will be connecting through old as long as everything going smoothly. And we will plan. If I change the system, I'll tell you what we do. Okay.
Yes, sir.
Okay. So Bablu also joined. So I will give him attendance. Bablu, are you joining or you are disconnected in between?
No, sir. Just now I joined, sir.
Just now joined. Okay, I have finished the class. Just I was discussing whatever left yesterday at neural network and protein-protein interaction network. It's not very difficult thing. You can read it. Since I have to cover the syllabus, so even easy things also I have to read up. Next class I will talk about computational medicine, which I have already shared with you that paper to all of you, paper authored by me. That paper you read, and 20th we are going to have exam. So maybe I'll discuss that paper on 21st with all of you. Okay. Okay. So I think next week we don't have any class. So you enjoy Puja holidays, and we will meet on 20th for exam.
Thank you, sir.
Okay.
Thank you, sir.
Thank you.
Sir?